Hello, and here we are again with the metabolism inside the mitochondrion. We discussed already the Krebs cycle and the preparation steps, and now we're going to focus on the electron transport chain. So let's begin. Okay, so we start here with the NADH from the Krebs cycle and glycolysis. This guy here is the complex number 1, and it is called NADH dehydrogenase. Okay, what is that? Basically, it's getting NADH and is creating NAD positive. In the process, flavin mononucleotide gets converted into its reduced form of flavin mononucleotide H2. So it's basically getting those electrons and giving them to the flavin mononucleotide. In the process, 4 protons are sent out from the matrix to the internal space. If we continue, we have the ubiquinone, which is another part of the electron transport chain, and this enzyme has mobility, and we will see then that appears in another part as well. Basically, inside there is a coenzyme Q10. This coenzyme Q gets the electrons from the flavin mononucleotide, and QH2 is created. This is the reduced form of the coenzyme Q. At the same time, on complex number 2, which is called succinate dehydrogenase, which is an enzyme involved in the Krebs cycle, if you remember, we get FADH2 from the Krebs cycle and glycolysis, and we create FAD. We donate those electrons to flavin mononucleotide to create its reduced form. Again, the ubiquinone here does the exact same thing. In fact, these two are the same. So here again, the coenzyme Q gets those electrons from the flavin mononucleotide reduced form, and QH2 is created, the reduced form of the enzyme, the coenzyme Q. Then we get to complex number 3, which is called Q cytochrome B oxyreductase. Okay, what is that? Q means from ubiquinone, cytochrome B to the cytochrome B, which is the center of this complex, oxyreductase. We're going to reduce the iron in here. So as you can see, those electrons are caught by the iron to go from iron 3 positive to iron 2 positive. And in the same process, the QH2 is converted into Q again. In this complex, we pump out protons as in the first one. 4 protons are popped out. Then we move on to cytochrome C. Cytochrome C does the exact same thing as the complex 3 and as the other steps. We just move electrons from one complex to another, from one enzyme to another. Here, the iron 3 positive is going to get those electrons from the iron 2 positive of the complex number 3, and we're going to create the reduced form of iron 2 positive. Then we continue and we get to complex number 4, which is cytochrome oxidase. In some literature, some books, they call it cytochrome C oxidase, because it's basically oxidating the cytochrome C, because it goes from iron 2 positive to iron 3 positive. this complex number 4 has the cytochrome A plus the A free. As you can see, there is cytochrome B, C, A and A free. So the exact same thing happens here. In complex number 4, we have another 2 protons going out. And then the last step is with oxygen. Have you heard in some book that oxygen is the last acceptor? Well, this is why. Because indeed he's going to get those electrons as the last guy and is going to be converted into water. The cool thing here is that this is like a hot potato game, because we start either on the complex number 1 or on the complex number 2, and we start giving electrons from here to here, from here to here. And it's just a game of giving electrons, passing them to the other enzyme until we get to the oxygen, which is the last acceptor. He's going to get all the electrons and he's going to create water. The purpose of this electron transport chain is to get protons out. As you can see here, the concentration of protons has increased. So once we have the protons out, the protons are going to come back in because a gradient is created, and they're going to get in with an interesting enzyme. This enzyme is called ATP synthase. It has 2 parts, the F1 and the F0. So the protons get here and they enter this channel and they get pumped in this way. As you can, if you follow the arrows, they get inside here and then they get in the matrix. When this is occurring, the rotor starts to rotate. As the rotor starts to rotate, the rod starts to rotate as well. And then this big complex of alpha and beta enzymes are going to create ATP, because they have both ADP and phosphate groups. Because of the rotation, these two get squeezed together and this creates ATP. Okay, so here we are with 4 protons. And why 4 protons? Because we need 4 protons to create 1 ATP. Because 1 proton is not enough, so we need more of them. And 4 are required to create 1 molecule of ATP. This is an interesting fact. If the protons come in the matrix via a channel, not using the ATP synthase, they are going to create heat and not ATP. ATP is going to be low and heat is going to be high. This is regulated by the thermogenin and is going to create thermogenesis. And that's how animals and some humans can actually get pretty warm in cold conditions. Okay, so if we watch what we have done, we pumped out protons in complex number 1, we didn't in complex number 2. We did in complex number 3 and as well in complex number 4. So there are 4 complex, well, if you count this as complex number 5, there are 5, and in complex number 1, 3 and 4 we pump out protons. How many? 4. 4 and 2. If we need 4 protons to get 1 ATP, here is the recap. NADH is going to pump out 10 protons because it starts with complex number 1. So it goes number 1, 4 protons, number 3, 4 protons, and number 4, 2 protons. And as 4 protons are required for creating 1 ATP, we can say that one molecule of NADH creates 2.5 ATP molecules. The FADH skips the complex number 1, and complex number 2 doesn't pump out protons, so we only get the protons out from complex number 3 and number 4, which are 6. And as 4 protons are needed for creating 1 ATP molecule, we can say that 1.5 molecules of ATP are created from 1 FADH2. Okay, so if we recap, 1 glucose molecule creates 30 to 32 ATP molecules, 10 NADHs from 1 glucose. Why is that? Well, 6 from the Krebs cycle, as well as 2 from the glycolysis and 2 from the transition step, so 10. Multiplied by 2.5, that's 25 ATP. 2 FADH2 from the Krebs cycle, multiplied by 1.5, that's 3 ATP, so we have 28. And the Krebs cycle, with the GTP creating ATP, is going to create 2 ATP, so we get 30. And if we count that glycolysis, the anaerobic glycolysis, also creates 2 ATP, then those are 32 ATP from 1 glucose molecule. So that's it for the mitochondrial metabolism. We already viewed what's inside the mitochondrion, how the metabolism works, and that's it. I will leave this image in the description below so you can download it and study it very well. You can share this video to other people to help them, and subscribe if you like the content, and see you on the next one. Hope you enjoy it.